Hello, and welcome to the Cell Press webinar on live cell imaging, focusing on cell-cell interactions, sponsored by Leica. Thanks for joining us. I'm Kara Lassen, Scientific Editor at Cell, and I'm here with my colleague, Ella Hinson, Scientific Editor at Cell Host and Microbe.
Today, our three speakers will tell you about their new findings on how cells interact and communicate. Cell communication is highly important in many diverse systems, and these talks will highlight the diversity of cell-cell interactions by examining cell-cell communication in neuronal responses, as well as in immune cell interactions during infection. Several of the advances that you're about to see are largely due to technology that allows us to track and manipulate living cells, providing us with a real-time glimpse of cell-cell interactions.
First up is Jeff Flickman from Harvard University. He'll explain the advantages of using live cells to explore neuronal connections. Next, we'll hear from Ellen Roby of the University of California at Berkeley. She will highlight how the immune system fights toxoplasma infection in the brain and lymph nodes. Our final speaker is Quentin Santow from the University of Oxford. He'll show how HIV exploits immune cell interactions to facilitate transmission.
Before we get started with the presentations, let's just go over a few practical points. Each of our speakers will talk for about 15 minutes, and then we'll have a question-and-answer session that covers all three presentations. If you have a question for any of the speakers, just click on the question tab at the top of the player and type it in. You can enter a question at any time, and there's no need to wait until the end of the presentations. Kara and I will present as many of your questions as we can during the Q&A session.
You'll see that there are three tabs on the viewer. One lets you provide feedback on the webinar, another has speaker bios, and the third provides technical support. If you experience any technical difficulties during the presentation, please click the tab labeled Support. Now let's hand things over to our first presenter, Jeff Flickman.
Hi, I am a developmental neurobiologist, and my interests are related to how the brain generates its wiring diagram. And one of the things we have discovered in looking at these wires that get set up in development is that interactions between nerve cells is a very important part of the way at least the mammalian nervous system decides which connections to hold on to and which to jettison in early life. I began in the 1980s studying these events in living animals, and because of that, at that time this was not a very common approach. There were many questions about why use living animals, and although now I think many people accept that for certain kinds of questions this is the way to go, there are many answers to the question why that I think are worth going through for people who may want to push into in situ imaging. So let me give you a couple of things to think about when you're talking about live imaging. For one thing, if you're imaging at the microscopic level, you'll be looking at cells, and cells themselves are, of course, living organisms, actually, inside an environment that's often the context of an animal. We see our work in an odd way, as if we are probing single-celled organisms inside an unusual domain, a weird pond for a protozoan, the brain being the pond and the neuron being the single-celled organism. And therefore, the kind of work we do is ethology at the cellular level, or if you think about cell behavior, cellular psychology. And if you think of people who study organisms, they certainly would argue if you want to watch the behavior of an organism, you've got to watch it over time. And if you want to watch cellular behavior, also it makes sense that one would have to do time-lapse imaging. But the obvious alternative is that if you're thinking about an event like development, which occurs in a relatively stereotyped way from one animal to the next, why not just use a series of static time points where one can get much higher resolution typically, and then just string together a series of single time points from different animals to generate a sense of how cells behave over time. And this seems like a reasonable approach, but for several reasons, the dynamics cannot be inferred from a time series composed of static time points from different cells. And one reason, of course, is that if the timing between different cells is not entirely synchronized, going from one animal to another and looking at a cell doesn't allow you to see how a single cell behaves. This is much the same if you're trying to understand the rules of a game like football, American football, where there's a lot of interactions. If you were doing it as a scientist who wanted to get the highest resolution data, you might go 1 second after the kickoff of a football game and take a really high resolution picture of a football game and then go back to your laboratory and study that for a week. And then next Sunday when another football game is starting, you wait till 2 seconds into that game and you take a picture, and then you bring that picture back and you study it. And you do that for every second until you have an entire, every second of a football game image, but no image is from the same game as any other, and try to infer the rules of a game like football. You'd find it very difficult, if not impossible. Whereas if you just sat in the bleachers and watched a single game from beginning to end, you'd probably learn enough that any future football game you went to, you'd understand. So that's one reason why one would like to do time-lapse. The second is that there are many events in development that are not monotonic. A monotonic event is something that, let's say, continues to get larger, or cells continue to divide, so the number of cells increases. But if you have things that wax and wane, get bigger and then get smaller again, you cannot infer that kind of data just by looking over time. And I'm going to just give you a couple of examples from my own work over the years from my colleagues and I, but where we have been looking over time and how we've inferred things that we couldn't see from single time points. So this following slide is a time-lapse image, time-lapse in the sense of 1 frame per day. Whoops, it went backwards, but if we go forward one more time, this in vivo imaging is an image of a neuromuscular junction in a living mouse. The red color you see there is a site on a muscle fiber that is contacted in development at postnatal day 8 in frame A by 2 nerve cells, a blue one and a yellow fluorescent protein expressing one. And we know that by postnatal day 15 or so, every neuromuscular junction in a mammal is only contacted by 1 axon. So we're absolutely certain that something is going to happen at some point between P8 and P15 or so that's going to change this from a multiply innervated neuromuscular junction to a single one. So the way we tried to understand this is to watch this over time. The questions we wanted to know is, for example, does one neuron take over the territory of another? Does that happen in one step or in multiple steps? How long does it take? And in these, we started by imaging every second or every minute in one day, and we found no changes that we could detect. So we finally found the right frame rate, and that is shown here, where we look at postnatal day 8, and we see the blue axon has slightly more territory than the yellow. And we come back at postnatal day 9, and we see the blue still has slightly more territory than the yellow. It's changed shape a little bit, but not much has changed. We come back later on postnatal day 9, and each time we re-anesthetize the animal, re-expose the muscle, re-image the same neuromuscular junction. We take another picture in postnatal day 9, still not much change. Then postnatal day 12, we come back, and we see, again, not a great deal of change, but in that area with the circle, the blue axon has invaded a little bit of the territory of the yellow axon, and then in the last time point at postnatal day 15, the blue axon has taken over all the territory previously occupied by the yellow axon, and the yellow axon, you can see that star at the very bottom is what's left. So that showed us things that we probably could not have inferred by single time point analysis. But this didn't tell us whether we could predict who won. It does look like the blue has slightly more territory than the yellow, so maybe that is why it's the ultimate winner. But here in another example, it is another junction that is multiply innervated, where blue has more territory than yellow. Whoops, went away. Just put that back. And then if we look at this at a second time point, we see that yellow 1 day later has taken over some territory, and at a third time point, yellow has more territory, and finally yellow, in this case, has won out over blue, and at postnatal day 15, only 1 axon is left in yellow. We could not have inferred that we couldn't predict who the winner would be by the areas they occupied early on by looking at single time points, but by looking at multiple time points, we saw that there was a complicated dynamic here. And we even found that it was not a monotonic change. So here's an example of an axon, the yellow axon, that has a small amount of territory on postnatal day 11 in panel C. In panel D, that axon has begun to invade the territory of the blue axon. But at panel E, at postnatal day 14, it has regressed, and at panel 18, the yellow is gone. That kind of behavior, that axons can come and add synaptic areas and then lose them, again, is impossible to see without dynamics. So for us, dynamics are very useful. So I just want to say a few words about some of the things I've learned about dynamic imaging that are worth keeping in mind, at least. And one is how often you should take images. Ideally, if you want to catch all the dynamism, you should use the equivalent of the Nyquist limit, where you oversample the dynamism by a factor of 2, just as you oversample the frequency in an image by a factor of 2 if you want to see all the resolution available. That's often difficult to do, but that will give you the full density of information that is available. And the second important question is where to do this. As an ethologist of cells, I would say you want to be as close to the natural habitat of these cells as possible. So in situ in a living animal is preferable. If that can't be done, and in many situations, of course, it can't be done, but you'll hear later today about looking at lymph nodes, and here in muscle or in brain, it sometimes can be done. One can take an explant and look at an explanted piece of tissue, and if that's not suitable, sometimes one has to resort to places where optical access is perfect, but the habitat is a little less native, and that would be tissue culture. Often, as I've said, these two goals can't be met in the same preparation, and one has to sacrifice one or the other to get the best possible information, or do both at different resolutions. I'm showing now another issue, which is just how many different ways one can image at different frame rates to get different kind of information. I hope now you are seeing in this movie, which should be coming up soon, the mitochondria moving inside axons that have been transgenically labeled by Thomas Miskeld and Martin Kirchensteiner and me when they worked in my laboratory. These are transgenic mitochondria moving in axons in situ, and the time lapse in this case required 1 frame per second. If we went much slower than that, although you would see that in each frame the mitochondria would be in different positions, you'd have a hard time tracking individual mitochondria because they're all labeled the same color. In this second example, which is a time-lapse watching nerve cells retract, but looking at the glial cell ensheathment, one sees 2 nerve cells associated with a neuromuscular junction on the left, but the only thing labeled here are the glial cells. And the top nerve cell is a dark double-bulbed object that is retracting, and the bottom axon that is staying there is that dark band going into the junction. In this case, 1 frame per minute was the appropriate time lapse to get the right data, and this is data from Darren Bishop and Thomas Miskeld. And then lastly, in this case, we were trying to visualize the way neuromuscular junctions enlarge. They get bigger as animals get older, but it keeps growing as animals grow over a period of a year. And the question was, do the neuromuscular junctions grow by adding branches or just by enlarging their branches? And in this case, each frame is a week. And Rita Ballasgordon and I looked over the lifetime of an animal, basically. And then I morphed those images together to give this sense of how neuromuscular junctions grow on muscle fibers. They just sort of add length the way a baby's hand gets to be an adult hand without adding fingers, just by making the fingers longer. Again, hard to imagine doing this in any other way. And then with live imaging, there's one last thing, and that if one is dealing with interactions between nerve cells or any kind of cell where the interactions are between cells of the same kind, one has to find ways of tagging each of the competitors if they're competing or interacting differently, despite the fact that molecularly they're of the same kind. And so we've been working at ways of giving sort of jerseys to the competitors that give them different colors to allow you to see them as separate, even though from a genetic or molecular standpoint it would be hard to separate them. And one approach is to take advantage of the many fluorescent proteins that are available. Mutations in a range of marine animals have provided really a remarkable spectral diversity of fluorescent proteins. It turns out that even 3 fluorescent proteins, a red fluorescent protein, a green one, a red one, and a blue fluorescent protein, is sufficient to generate all the colors a human can see, just the same way that black and white television became color television with 3 phosphors, a red, a green, and a blue phosphor. And that's because we only have 3 photoreceptor types in our eye, a red one, a green one, and a blue one. And so we mix various amounts of red, green, and blue to give us all these colors. And so that's what we did. This is work that Jean Levet did, and I won't go through the details here. This has been published a while back now. But it's basically generating transgenes that through recombination, a single transgene can either be red, green, or blue. And if a single copy of this transgene is inserted into an animal, then you will find in the brain, for example, in this case, astrocytes that are either red, green, or blue. And you can see immediately that 3 colors is more than one, but not enough to really tell you where every cell is. For example, all those red areas are probably many cells that are just the same color but are separate. So if you want to see the boundary of every cell, you would need more colors than just 3. It turns out when you make a transgenic animal, a insert like this is often inserted in tandem in many copies. And each of those copies randomly will choose one of the 3 colors to generate. So in collaboration with Josh Sains and Jean Levet, we developed mice like this where, for example, one neuron might have 3 doses of the red fluorescent protein, one of the green and one of the blue, and that cell might look a brownish-orange. Another cell, just by chance, through this recombination, might end up with a blue, a red, and 3 greens and end up light green. So these cells are molecularly not different, but in fact they end up with different colors. And then the brain is filled with cells of many different colors. So this is the hippocampus mainly, what you're seeing in the middle. At the top is the cerebral cortex. And if we zoom up on the dentate gyrus, you just see lots of cells and obviously more than 3 colors. We get about 100 colors here that we can discriminate. It's largely related to the quality of our imaging and how many colors we can see. Finally, it's important to realize that those 3 colors is all we needed to generate all those colors. You take a blue image, a green image, and a red image, and you superimpose them, and you generate these large number of colors. Let me just go back again here. This large number of colors. And these are now new generation mice that Dawin Kay, again in collaboration with Josh Sains, we've made in which all the colors are in the membranes of neurons rather than in the cytoplasm. That's a huge advantage because it means now no matter how big or small the process is, you have the same amount of fluorescence per square micron, so the finest processes are easier to see. Also in the newest lines of mice that we are making, each of the fluorescent proteins is coming from a different marine animal, allowing us to make secondary antibodies to each of these colors and then stain with antibodies of different Alexa dyes to each of these fluorescent proteins to give us very bright labeling for fixed tissue. And then in this last slide, I'll just show you what it looks like with a confocal microscope if you then image in on the terminals of these axons going out in the periphery. These axons, by the way, being labeled like this is very similar to this phenomenon, which humans also developed, in this case, to understand how the wires in thinking machines, computers, are connected from one place to another. So this is some form of convergent evolution, I guess. And here is the final example, just showing that because of the colors now, I can say with absolute certainty, for example, that the 2 purple-colored neuromuscular junctions on invisible cylindrical muscle fibers running up and down are from the same neuron, and with equal certainty, I can say that the orange neuromuscular junction or the greenish-blue one or the light blue one are from different neurons, even though those neurons are maybe a centimeter away. So color is a very powerful way to allow us to look at the wiring diagram and the connectional maps in the peripheral nervous system. That's really all I have to say. Thank you very much.
Thanks, Jeff. And now moving on to the immune system with Ellen Roby. Ellen?
Thanks. So I'd like to tell you about work from my lab using two-photon microscopy to examine cell-cell interactions between immune cells and parasites during in vivo infection. So host pathogen interactions normally take place in tissue environments such as lymph nodes that are highly organized and tightly packed with cells. However, we know very little about how pathogens and immune cells interact with each other during infection. And the combination of two-photon microscopy together with genetic methods for in vivo labeling, such as discussed by Dr. Lichtman, together provide an important tool to address these questions. So the method is similar to confocal microscopy, but uses a laser that delivers intense, short pulses of light that allow for excitation at long wavelengths and limit excitation to the focal plane, as shown in this image. And the result is deeper penetration into tissue and reduced toxicity, phototoxicity, and that makes this a really great method for live cell imaging within relatively thick tissue samples. So direct visualization is important because it tells us where, when, and how pathogens and host cells interact with each other in vivo. And that's especially important for T cells, which are cells of the adaptive immune system that have to directly contact antigen-bearing host cells in order to recognize pathogens. And in addition to detecting the pathogens by directly contacting infected host cells, this direct contact also allows for directed delivery of protective cytokines and thus makes the adaptive immune response more effective. Now, in principle, these processes can be studied in vitro. However, this approach misses the role of important cell types that are difficult to isolate and culture in vitro. And an example that's relevant to my talk are these specialized macrophages that line the subcapsular sinus of the lymph node, and they're revealed here by this marker that they express, CD169. These cells have really interesting and unique functions. They're very different from, for example, bone marrow-derived dendritic cells, and they're very difficult to study in vitro. Now, the pathogen that we're studying is a protozoan parasite related to malaria. It establishes chronic infection in the host by forming cysts within the brain, generally asymptomatic. Its toxoplasma is a generalist. It can invade any nucleated cell in a wide variety of warm-blooded hosts, including humans. And it's estimated that about 30% of people worldwide are infected with toxoplasma, the vast majority without any symptoms. Toxoplasma is an intracellular pathogen, so it invades host cells by an active invasion process, forms a specialized intracellular compartment called the parasitophorous vacuole, and that protects the parasite from the host degradative machinery and allows it to recruit goodies from the host cell, including attachment with the host ER. When parasites are inside the parasitophorous vacuole, they can replicate, and eventually the parasites lyse the cell and egress as motile parasites, which can then evade another host cell. And some of the reasons that we love toxoplasma are that it's a genetically tractable organism, it has really interesting biology, and the mouse is a natural host for toxoplasma, and the in vivo toxoplasma mouse infection model provides a really great system to understand how mammals control intracellular infections. So CD8 T cells are the most important branch of the adaptive immune response to toxoplasma. However, the parasite, when it forms this parasitophorous vacuole or cyst wall, this structure poses a barrier for the parasite antigens getting into the MHC class 1 presentation pathway and makes it difficult for those antigens to be seen by CD8 T cells. And so that poses a number of questions of how the CD8 T cells actually detect the parasite antigens in vivo. What types of antigen-presenting cells are involved? Do those cells need to be directly invaded by the parasites, or can bystander cells present, etc.? And also, what happens to parasites when an effector T cell interacts with an invaded host cell? And do T cells use different modes of parasite detection and delivery of cytokines during different phases of the infection cycle? So the system that we've used to study this is cartooned here. We're using parasites that have been engineered to express red fluorescent protein, and we're infecting mice that have antigen-specific parasite-specific T cells that are labeled, and also the hosts express cell-type specific labels, fluorescent labels for various host cells that are of interest. And then we image different tissues of the mice, and I'll be talking about lymph node and brain by two-photon microscopy, and of course using other support methods such as flow cytometry and confocal microscopy. So the data sets are four-dimensional data sets, X, Y, Z, and time, and the images are then rendered to produce three-dimensional images, and then cells can be tracked and quantified as they move through these imaging volumes to determine motility and cell-cell interactions. So this is an image, this is a single two-photon movie rotated on the top here to show you the three-dimensionality of the data, and on the bottom, the same imaging data set is projected along the z-axis and played as a time-lapse sequence sped up about 200 times over real time. This is actually an example from the other half of my lab that studies the thymus. And the two points I want to make here are, one, that when you look at these images, you need to keep in mind that although the volume looks like it's free of cells and relatively spacious, the tissue is actually tightly packed with cells, and as shown by the scanning electron micrograph of the tissue to the left. And you can see the lines represent the tracts of cells as we follow them through the imaging volume. And all of these images I'm going to show you are played in a loop and represent about 20 minutes of imaging time, so they're sped up about 200 times over real time. So we need to be able to follow parasite-specific T cells in vivo. However, the endogenous parasite antigens that are recognized by T cells are just beginning to be discovered by our group as well as others. So what we've done instead is to use a parasite strain which is engineered to express a model antigen, chicken ovalbumin. These were generated in the lab of our collaborator, Boris Strypen. And the parasite actually secretes the ovalbumin protein into the parasitophorus vacuole, where the protein can eventually make its way to the cell surface to be recognized by a CD8 T cell in an infected cell. We're also using engineered T cells, which are designed to express a fixed T cell antigen receptor that recognizes the ovalbumin peptide presented by the host MHC molecule. And this allows us to follow antigen-specific, parasite-specific T cells in vivo. Now, when we infect mice with the fluorescent parasites, we often find the parasites within these specialized macrophages at the lymph node subcapsular sinus that I mentioned earlier. And these are found in this region about a few hours after ear flap inoculation or several days after oral infection. And work from our labs as well as a number of others has focused attention on this subcapsular region of the lymph node and these specialized macrophages that reside there for their role in trapping lymph-borne pathogens and initiating immune responses. So this slide shows a two-photon data set collected in the lymph node of a mouse that has been recently infected with red toxoplasma. And the memory T cells are shown in yellow, the collagen of the capsule is shown in blue, the macrophages in orange, and the parasites in red. And on the left, you see a single time frame from the movie projected along the y-axis. And what we've done here is just take a slice through the top, the middle, and the bottom of the Z-stack, and then project those and display them as time-lapse sequences to the right. And in this view, I hope you can appreciate that within this single imaging volume, there are three distinct regions where T cells are forming low motility clusters around parasite-infected cells. Now, in this view, we're seeing the, let's see, to advance this here. Oh. So in this view, on the left, you see a cluster of memory T cells surrounding a parasite-infected cell in which the parasites express the ovalbumin protein. And these clusters only form when there's an antigen-specific interaction between the T cells and the parasite-infected cells. So in the right-hand movie, where the parasites don't express ovalbumin, you see that the memory T cells still go to this region of the lymph node and migrate near the macrophages, but don't make stable contacts. And here's another example. In this case, we occasionally see these examples where rather than interacting with these resident non-motile macrophages, the T cells can sometimes form stable contacts with motile infected cells. And in that case, the T cells get dragged along in the lymph node behind the infected cell. In these examples, the infected host cell is not labeled, but we can see it's there because of the parasite and the T cell following along behind. Now, we occasionally see T cells, we see the parasites leaving the host cell, egressing from the host cell. And when this occurs, we often see the parasites entering one of the memory T cells, directly invading the T cells from inside the contact. And so that's shown here in this series of still images, where in the first image you see the parasite inside, surrounded by a cluster of T cells. And then at 90 seconds into the movie, the cluster begins to break up, and the parasite goes from inside the cluster into one of the memory T cells that was originally part of that cluster. And here's another example shown both as a time-lapse sequence and also as a video where the time-lapse sequences came from. And you can see in the beginning of the movie, which is playing as a loop, the parasites, there are two parasites inside a non-labeled host cell surrounded by 3 or 4 T cells, and then the parasites then leave the infected host cell and enter one of the memory T cells. So how does this compare to T cell detection in the brain? So to look at this, we've used a similar system where we have a labeled ovalbumin-specific T cells, but now in this case we've infected the mice with a low dose of parasites expressing RFP and ovalbumin, and we wait several weeks to establish chronic infection and then perform 2-photon imaging of vibrotome-cut brain slices. And so some of the questions we're interested in here are, again, how do the CD8 T cells actually detect the parasite in this setting? And also, can the CD8 T cells see the parasites inside cysts? It's known that cysts reside within intact cells in the brain, mostly in neurons, and so there's the potential for parasite antigens to be presented to T cells from a cyst-containing host cell. However, when we look in the brains of these mice, we see that there are very few T cells surrounding isolated cysts, and that's in spite of the fact that there's plenty of ovalbumin inside these cysts. So the parasites are making ovalbumin, and they're secreting it into the cysts, but the T cells are completely ignorant of this. And instead, we see T cells clustered in regions of the brain where we see single isolated parasites, but not intact cysts. And this is a time-lapse image showing the T cells. These are the ovalbumin-specific T cells actively migrating around in the brain parenchyma in the vicinity of these two cysts, shown in sort of the middle left, upper left part of the movie. And the T cells are vigorously migrating through the brain parenchyma in the vicinity of the cysts, but not making any contact with the intact cysts. In contrast, we do see these interactions with these individual parasites, which are in the form of granuloma-like structures, aggregates of macrophages, which contain the isolated parasites, and that's where the T cells are congregating. And here's a time-lapse image to show you how these interactions look. Now the T cells are in green, the macrophage-like cells are shown in blue, and the parasites are in red. And you see that the T cells contact the macrophages that make up this aggregate, and they slow and pause when they're in contact with these granuloma-like structures, but they don't actually appear to directly contact the parasites within these granulomas. So to conclude, we've seen that CD8 T cells form stable interactions with invaded host cells, with invaded lymph node subcapsular macrophages during acute infection, and that the direct contact with parasite-containing host cells exposes the T cells to invasion by the parasites. In contrast, in the brain, T cells form granuloma-like contacts with granuloma-like aggregates of macrophages, relatively motile contacts, and ignore parasite-containing cysts. And these two distinct modes of recognition by CD8 T cells are revealed by 2-photon time-lapse imaging in situ. And this is just to mention the many people that contributed to this work, folks from my lab, as well as collaborators who made critical contributions by providing Toxoplasma strains and mouse reporter strains. So that's all I have for now.
Thanks, Ellen. And now on to our final speaker, Quentin Satentau, who will discuss how HIV can hijack some of these cellular interactions in the immune system.
Thank you very much. So my lab in Oxford has been working on how HIV-1, the human immunodeficiency virus, which is the cause of AIDS, moves between cells that are permissive for the virus. And these are cells of the immune system that express viral receptors that allow the virus to enter and replicate inside the cells and ultimately cause disease. The classical paradigm of how viruses spread between cells is that infected cells will release virus particles, and those virus particles will diffuse in the fluid phase randomly until they meet a receptor-bearing target cell, at which point they'll infect that cell. This is a rather inefficient process because the virus may have to diffuse a great distance in order to find a target cell, or it may be in a very dense tissue where it may have trouble diffusing and it may not meet a target cell. And in the case of HIV, the average life cycle of the virus from being released to finding a new target cell in culture to that cell producing new particles is about 24 hours. However, there's another mode of direct virus spread between cells, and that is when the cells contact each other, an infected cell can pass virus directly into the uninfected receptor-bearing cell. This has been known for some time for a number of viruses and was first described for retroviruses, of which HIV is a member, about 10 years ago. Now, the life cycle of HIV spread directly between cells is approximately twice as quick, although that varies between virus strains and cell types. And that clearly shows that it would be to the virus's advantage to spread in this directed manner between cells. So I'm going to talk about three things in this presentation. The first is something that we've called and others have called a virological synapse. The second is membrane nanotubes, which are actin-containing tubes that form between different cell types and allow virus transmission. And the third is a new form of viral cell-to-cell spread, which is used by HIV to infect macrophages. Coming to the virological synapse, this is work done by Claire Jolly and Sonia Welsh. This shows an electron micrograph, and on the left is a low-power image. At the top, you can see an infected T lymphocyte, and at the bottom is a target T lymphocyte that has CD4 and a co-receptor for HIV-1. On the right is a three-dimensional reconstruction of that electron microscopic image by a technique known as tomography. And what it shows is that the infected T cell has virus budding in red from its tip where it's contacting the uninfected target T cell. And the virus is budding at that polarized tip and infecting the target cell. The other feature of this virological synapse is that you can see there's a large number of organelles, cellular organelles, also polarized with the viral budding, including mitochondria, lipid bodies, secretory lysosomes, and the microtubule organizing center. And what this represents is polarization of the cytoskeleton in the infected cell down towards the site of contact with the receptor-bearing target cell. These two cells are held together by adhesive interactions, mainly by integrins, and actin and microtubules focus the secretory machinery down to this interface, and the virus moves across. I want to show an old movie now, which was one that we made really right at the start of our investigation into the virological synapse, and this was done by Yvonne Mitar in my lab. In the top, you will see a CD4 T cell that is expressing CD4 fused to green fluorescent protein, so it fluoresces green. and in the bottom is an HIV-positive T cell, and the HIV surface glycoproteins are labeled with a specific antibody that's coupled to a fluorochrome in the form of a Q dot, and these are crystals that emit at certain fluorescent wavelengths, and the advantage of them is that they don't fade over time. So the movie will now run, and you'll see this contact time. This is a time-lapse movie over 1 hour, and what this shows is that these conjugates between infected and uninfected cells are stable for at least an hour in many cases. The other thing to note is that the GFP has bleached by the end of the movie, whereas the Q dots haven't. We wanted to look more closely at synapse lifetimes and try and understand how long it took for HIV to move between an infected and an uninfected cell. And to do this, we again labeled infected cells this time in red and the target cell in green. And we used some image processing to expand the contact zone so that we could see it more easily and process it with image processing software. And that then allowed us to run a live cell movie of the contact sites between the red and the green cells. And this was then binarized to show the contact sites in isolation. And you can see the contact sites coming and going as conjugates form and break apart over time. And using that information, we were able to work out the different lifetimes of virological synapses. And you can see that the vast majority are relatively short. The average virological synapse lasts for about an hour. And then there's a few events that last up to about 5 hours or possibly more. And we assume that it's the longer events above an hour that are the ones that lead to viral transfer and infection of the target cell. I want to move on to the second type of transfer, which is related, and this concerns the formation of what have been called membrane nanotubes, and this was work done in collaboration with Steffi Sawinski and Dan Davis at Imperial College. When T cells contact one another, and in this diagram you can see one in red, one in green, and then pull apart, often they maintain a connection between themselves in the form of a membrane-bound tube that contains actin. And that tube can be as long as 100 microns or even, in some cases, longer. And interestingly, that connection at one of the cell ends often has a mini kind of synapse, which means that the cytoplasm is not continuous between the two cells, but there's a barrier at one end. The movie I'm going to show next is the formation of one of these nanotubes as the two T cells pull apart. And you should see the two T cells at the top, red and green, and as they extrude out, they pull out a nanotube. The next movie will show a nanotube that's already formed, and what you'll see as it rotates is that there are blebs or bulges in or on the nanotube, and this is material or cargo that's being carried along the nanotube in a directional manner from one cell to the next. So we asked ourselves whether this cargo could potentially be virus and whether viruses such as HIV could use nanotube-like structures to travel between distant cells. So the setup was very similar. T cells were labeled with HIV fused to GFP, so infected cells are green, and then the target cells are labeled in red. The movie is a series of, there's no movie here, it's time-lapse images. On the left-hand panel at 0 seconds, there's a nanotube between the green cell at the top and the red cell at the bottom, and in the white box one can see virus that has moved from the green cell and is about to contact the red cell. And then in the third image at 40 seconds, the virus has contacted the T cell. And in the next set of blow-ups of that sequence, you can see at 0 seconds the virus is about to contact the cell, and at 65 seconds in the fourth panel, the virus appears to have fused with the target cell membrane. So this does appear to be a mechanism for long-distance virus spread between lymphocytes and potentially other immune cells. I want to now briefly talk about some work by Walter Mothers at Yale, who works on a related retrovirus, another retrovirus called murine leukemia virus, which infects cells such as fibroblasts. He's shown a similar phenomenon, but in a way more dramatic. And the system is, as shown here, that if one takes an infected cell and that then contacts an uninfected cell that bears the receptors, there's a process where the infected cell joins with the receptors on the uninfected cell using the viral envelope proteins to trap the receptors. And then when the cells move apart, they form membrane tubes, also known as filopodia. And viruses can move along those using actin-based motility to infect the target cell very efficiently. And this is the movie, and you should see virus moving in red from the infected cell to the target cell. And as the cells pull apart, so they extrude the filopodia, and the virus particles continue to bud from the infected cell and move along the filopodia. And the final frame shows the total number of events of virus transfer, and one can appreciate the massive infection that takes place by this form of transfer. The final model that I want to mention is HIV-1 spread from T cells to macrophages, and that's work that's gone on in my lab. And the movie I'll show is made by Amy Baxter in the lab. And what it shows is T cells that have, again, been labeled with GFP fused to HIV. So the T cells that are infected are green. And there are 3 T cells in the movie that will be taken up by a single macrophage. The first T cell at the bottom is taken up. Then the next T cell is engulfed. And then there's a third T cell at the top, which the macrophage seeks out, wraps some membrane around, and engulfs. And the T cell is then taken up, and there's a macrophage with 3 HIV-infected T cells in it. And I think what's interesting is that the macrophage ignores all the uninfected T cells surrounding it that are not green, and only goes for the infected ones. Now, when the macrophage has engulfed the T cells, the macrophage becomes infected. And this panel shows an engulfed T cell that's budding virus from one of its tips. If one looks at panel C, you can see in this tomogram the red virus particles leaving the T cell at one pole, and those are then entering the macrophage. And that will infect the macrophage, as shown in this final image from Chris Duncan. This is a few days after macrophages have engulfed T cells, and they're robustly infected, as shown by the green label. So to conclude, we find that live cell imaging is really essential, as the other 2 presenters have mentioned, for probing dynamic interactions between cells. And without it, we wouldn't be able to understand things like velocity of movement and function of things like nanotubes and virological synapses. If it's coupled with high-resolution static imaging, such as electron microscopy and electron tomography, one can really get to grips with structure-function relationships, both in cell-cell interactions and in virus-cell interactions. And what I've shown today are all in vitro live cell models and movies. What we'd really like to do is move, as in the previous 2 presentations, towards in situ following of HIV movement in living tissue. And I'd just like to acknowledge the people that did the work in my lab at University College, Imperial College in Heidelberg and Yale, and our funders. Thank you very much.
Thanks, Quentin. We now can take some questions from our listeners. And first up, we have a question for Jeff from one of our listeners who wants to know if you can use the Brainbow system to also label single cells for in vitro use.
Yes and no. You certainly can label cells in culture. But if the cells are already in culture, the transfection has to be with a promoter that expresses well in the cell in culture. And Thy1, for example, which works very well in animals, most neurons in culture, when we transfect with Thy1, the expression is slow to turn on. So although there's nothing wrong with the idea, one has to pick the right promoter to get it to express. The other thing, of course, is that fluorescent proteins take a little bit of time to turn on, and so one has to calculate that in. One way to deal with this is to transfect an animal or have a transgenic animal and then create the culture out of cells that are already expressing, and then one can do rainbow imaging in culture.
Thanks, Jeff. Our next question is for Ellen Roby. We're curious, since you're showing cells labeled with GFP and YFP, and there's a lot of data that looks at co-localization and cell migration patterns, how do you monitor direct contact instead of co-localization and migration patterns? And this listener also wants to know what you think about GRASP or BLINK technologies.
So for the first part, in terms of the monitoring cell-cell interactions, I actually didn't present any of that data in my presentation. But the way we've done that in the past is if you have two different cells which are labeled with different fluorochromes, and it's not a big problem if there's spectral overlap as long as they're on different cells. Then we've developed methods to define the outline of the cells and to measure the distances between either the edge of the cell or the centers of cells. And I believe some of the commercial software packages for image analysis also include those kinds of quantification options. With regard to the GRASP, I'm actually not familiar with that method. So maybe I don't know if any of the other speakers could speak to that question.
I mean, this is an approach where a protein, fluorescent protein, is more or less made by two pieces that, if they get close enough together, can form a single fluorescent protein. So it has in common with techniques like FRET, it's a measure of things that are very close together, and it's being used in the nervous system often, not that often so far, but in C. elegans, Corey Bargmann's lab has used it, for example, to see where synapses occur between two nerve cells by generating a fluorescent protein in between.
And maybe I would just add that I think that the issues about co-localization when you're talking about things within the cell, it sounds like that's the kind of issue where this GRASP technology would probably be useful for looking at cell-cell interactions. There are different kinds of approaches you need to use.
Clinton, we have a question for you. We have a listener who wants to know, what is the role that actin and microtubules play in the three types of HIV cell-to-cell spreading that you showed?
Well, yep, they are central to all three types of cell-to-cell spreading. If I start at the back, phagocytosis by phagocytes requires the actin cytoskeleton to be completely functional, and of course the process requires the formation of a phagocytic cup and then the closing of that cup. And if one blocks actin remodeling with inhibitors, one completely prevents the uptake of these T cells. Similarly, virological synapses can be completely prevented by inhibitors of actin remodeling. The microtubule organizing center and microtubules are also important in the virological synapse for polarizing viral production to the point of contact between the cells. And finally, the nanotubes contain actin, and so if one paralyzes the actin cytoskeleton, they won't form. So in each three case, it's absolutely essential that the cytoskeleton is functional.
Great. Our next question is for Jeff. There's a couple of people who want to know how you prevent and avoid cell death or photoleaching when you're doing this time lapse, and what kind of laser power you would use to make sure the cells are unharmed.
Right. I think this is a question that all three of us are very familiar with. One of the fundamental problems with fluorescence is that there is often a byproduct of fluorescence, which is generating from the triplet state, singlet oxygens that ultimately give rise to reactive species that can cause serious damage to cells or synapses. As a rule of thumb, I would say, first of all, if the image looks really good, if a single frame of your time-lapse looks really good, you're using way too much light. You want to bring that light level down to the point where the image is maybe a bit noisier than you would like. It's still okay because by time-lapse, that noise will be random between frames, and it usually does not get in the way of interpretation. But if you're getting enough light to get beautiful images, almost certainly, sadly, you're using too much. As you've already heard, two-photon is a better technique for bleaching than confocal because you're only bleaching in one plane. Generally, wide-field imaging is better than scanning imaging because the intensities when using is lower. But what we do in my lab is we really titrate that light down and increase the sensitivity of our detectors to a point where things are comfortable. And so there are new avalanche photodiodes, and there are these gallium arsenide photodetectors that are much more sensitive for imaging than the traditional kinds of detectors, and those help us a lot. But we typically try to keep our imaging down below 100, a 0.1, under a milliwatt, maybe more like 0.1 milliwatts or even less in our scanning approaches.
And maybe I just add one thing to that, which is that there's, like Jeff said, you always want to be using the minimum laser power, and you're always compromising, at least in the kinds of experiments we're doing, between how bright your floor is. You know, there are many informative labels you'd like to look at, but you need a fairly bright label to be able to see it within the tissue. How often you sample when you're doing a time-lapse series, and how deep you're going in the tissue. That there's always compromises involved in that, and that laser power is, of course, an important part of that.
Ellen, when you inject the labeled antigen into mice, how long are you able to visualize that color in vivo?
So in the case of some of the data I showed where we're injecting fluorescently labeled antibodies, that's kind of a special case. That's a case where the antibody is sort of directly delivered to the area that we want it to go. In that case, the antibody we can see that for several hours. If you do experiments where you load cells with a fluorescent dye, then those dyes will last for a few days, depending on how much cell division. And of course, one of the big advantages of the genetically encoded reporters, GFP and the like, is that they remain in the cell the whole time.
And Quentin, so you showed a macrophage ingesting T cells. Can those viruses then go on to infect other cells the way that the T cell was infected?
Yeah, absolutely. One of the things about macrophages is that they are readily infectable by HIV, as I showed. Unlike T cells, however, macrophages don't die when they get infected, or at least they don't die quickly. When CD4 T cells get infected by HIV-1, they tend to only live for a day or two, and we know that's both in culture and in the body. So they don't have long to produce new virus. By contrast, macrophages, we believe, can probably live for weeks or months, having been infected by HIV, and they will then seed virus. They will produce small amounts of virus that can then go on to infect other T cells, other macrophages, and potentially other cell types such as astrocytes in the brain. So HIV-infected macrophages are what we would call a viral reservoir, and it's really important to try and understand how we can either prevent or reduce infection of the macrophage.
And Jeff, what software do you use for your rainbow techniques?
We generally don't use special software for that. Most of the images that are taken with confocal only need a program that will combine the three channels. And to cover that point just slightly more, it's important that the three color channels we use are from fluorescent proteins that do not overlap. So although GFP and YFP are very easily used fluorescent proteins, it's very hard to get a clear separation of them. So we typically use a blue fluorescent protein like cyan fluorescent protein, something like yellow fluorescent protein, and then something in the red. And then we don't simultaneously image all three colors, but image the blue and the red color simultaneously, and then in a second image do the one in the middle. And that way we get very little overlap. And then in any program, Adobe Photoshop, for example, one can just mix those three colors as different layers and get the full color image.
And Ellen, you touched on this briefly when talking about some of the trade-offs in imaging, but what do you think are some of the advantages or disadvantages of using two-photon as opposed to confocal or some other techniques?
Well, I guess for confocal, it certainly has a higher resolution than two-photon, and it also allows you to use a variety of different fluorescent antibodies to identify cell types. It's generally not as good for time-lapse, in part because of the extra photo damage that you get with the lasers. And also the penetration with confocal into the tissue is limited to around 50 microns, where you can go hundreds of microns with the confocal. I guess those are probably the main ones I can think of.
For us, the main problem with two-photon is that when you want to do three colors simultaneously, you have to switch the wavelength band of the two-photon, and that's time-consuming. So it's very hard to get 3D stacks with two-photon in three colors, at least when we try it. So we have to use confocal.
Yes, I think we get 4 or 5 colors simultaneously, and I guess it probably depends on just having floors that are separated enough to start with. Mm-hmm. And I know that people are starting to also have 2 simultaneous lasers tuned to different wavelengths.
Right, although that's very expensive.
Yeah, that's true. Some of the new lasers also will switch between wavelengths fairly quickly.
Yes.
And Quentin, why are HIV-infected cells phagocytosed by macrophage? Is apoptosis of the infected cell involved in this at all?
Yes, we are currently grappling with that question. The answer, I'm pretty sure, is yes. The majority of HIV-infected T cells that are engulfed by macrophages do appear to be either apoptotic or post-apoptotic, otherwise known as necrotic. So it seems that the signals that macrophages are reacting to are not HIV-specific signals, but the fact that the infected T cells are dying. And, of course, the job of macrophages, or one of the jobs of macrophages, is to clear those dying cells up.
So as we're beginning to run out of time, I just had a question for all 3 speakers, and we'll probably start with Jeff. But I'm curious what you think the major problems or impediments there are to improving live cell imaging in the future. And do you think it's fluorescent proteins or microscopes or lasers, or where do you think the technology is going, and what's sort of limiting at this point?
Well, there is a fundamental limitation with traditional optical microscopy, which is the diffraction limit. So very interesting phenomena that occur below the resolution limit are hard to see, and that limit is around 200, 250 nanometers. And as I'm sure many of your listeners know, there are a wide suite of new super-resolution techniques that go through and break through this diffraction barrier. It is going to be a challenge to figure out how to use those in situ. Some of them take a long time to get an image that, in the sense that they're iterative methods. So one challenge is breaking through the diffraction limit, which now can be done, but in living animals, so far that has been somewhat challenging, I would say, to do. A second, I think, general problem is depth. One wants to look much deeper into animals than one can get even with a two-photon microscope. One's talking, the limit is probably around a millimeter at the best-case scenario. And how one gets deeper, there are many ideas, and people have come up with little probes that are gradient lenses that allow you to stick a probe in and look at a single area deeper. But one would like to think about techniques that go deeper, and that's a technology that is in the interest of many people, and I think we're going to see some progress in that direction as well.
Thanks. And Ellen, do you have something to add?
Yeah, well, I agree with what you said, Jeff, and I think also the biological systems are I think, a challenge. Finding ways to introduce informative fluorescent labels without perturbing this tissue and the events that you're trying to study, better readouts for intracellular events, signaling events, et cetera, and methods for combining perturbations of individual molecules or processes with monitoring readouts. I think the biological systems are lagging behind in some cases for the questions people would like to address.
And Quentin, the same question to you.
I can't add anything to the discussion on new technology. All I can say is what I mentioned in my presentation, which is that what we would love to do is get some of the technology that Jeff and Ellen already have into a containment 3 laboratory. I'm not sure that anyone has that kind of kit available for looking at category 3 microbes such as HIV.
All right. Well, with that, we've come to the end of our session. If you missed anything during the webinar or would like to listen to it again, a recording will be available shortly after at the same site. Be sure that you registered so that you'll receive notification as soon as it becomes available. We'd like to take this opportunity to say thank you again to all of our speakers, Jeff, Ellen, and Quentin, for a very interesting set of presentations, and thanks for answering all of our questions. We're also very grateful to our sponsor, Leica, for their contributions that made this webinar possible, and thanks to our host, Bright Talk.
And finally, we'd like to thank you for tuning in today to learn more about live-cell imaging. Keep your eyes and ears open for more Cell Press webinars on cutting-edge topics in biomedical research. If you have any comments about this webinar specifically, or if you have suggestions for future topics, we'd love to hear from you, and you can email us at cpwebinars@cell.com. We hope you've enjoyed this webinar, and thank you again for joining us.